Can somebody confirm that you can hear me?
I can hear you.
Thank you. All right. Before we begin, are there any questions? I should mention that I've done some grading. I only graded one of the labs. I'm not sharing my screen, I don't think. So let me share. Let me look in my grade book here. I think I only graded one of the labs for last week, which means on that lab, which I think is lab one, maybe? No, it would be lab two, sorry. Um, I got worksheet for lecture two graded last week. Actually, I got worksheet for lab 00 graded a second time. And that's rare that I get something graded a second time this early in the term. Worksheet for lab two has been graded the first time. That means if you did not get 100%, then you can submit corrections by this Saturday. I'm not sure when I will get worksheet for lab three graded the first time. I doubt I'll be able to finish every one tonight, but if I do, then you would have to submit corrections by this Saturday. If I don't finish every one tonight, which will likely be the case, I might be able to get half of you done, but I doubt I'll get all of you done. Then it will be later, probably not this Saturday, but a week from this Saturday, that you may turn in corrections. Any questions about any of that? All right. Let me shut down the grade book. Go to home. And then let me call up the lab module. Oops, we're still making the, that's why my computer is so slow. They're still making the lecture. So, oh, I need to share my screen. Oops, wrong one. That's why I went there. There we go. All right, today we're going to be discussing Lab 4 on stains. You should also read the unknown project file instructions on Canvas for next week. I'm going to be talking about the unknown project on Tuesday. And you can find that. I think it's week 5 of the lab. You need to read this one here, the unknown instructions.docx. And I'll talk about that next Tuesday, the unknown project. Any question about any of that? So lab 4, that's what I wanted. Oop, missed it. So lab 4 stains. Oh, good. The lecture file finished. Now my computer will start working right. Lab 4 is on staining techniques. Know that there are simple stains, differential stains, and special stains. You do need to read the textbook, especially on this one. I should read page 73 to 75 on size, shape, and arrangement of bacterial cells. Read about the bacterial cell wall on page 80 to page 83. Read about the simple stain on page 65, and then read about the differential stain, the Gram stain on page 65 to 66, and the acid-fast stain on page 66 to page 67. And then read about special stains, page 67 to 68, the negative stain, staining for capsules, which is on page 67, and then page 75 to 77, the endospore stain on page 68, in page 92 to 94, and then the flagella stain on page 68 and page 77 to 78. In today's lab, there will be some video clips and a virtual lab. Your learning objectives for today: 1, be able to prepare a bacterial smear for staining, and please understand what the bacterial smear is. It is what you have to do before you do the staining. It is its own steps separate from the staining. And then 2, understand the principles and procedures used when performing the simple stain, the differential stain, and the special staining techniques. Oops, I just lost it. 3, interpret the results for the simple stain, a differential stain, and a special stain. And then 4, be able to define the following terms. What's a simple stain, a differential stain, a special stain? Coccus, Bacillus, beryllium, peptidoglycan, gram-positive cell, gram-negative cell, acid-fast stain, primary stain, what's a mordant, decolorizer, counter stain, what's a capsule, endospore, and flagella. Any questions about any of that? So bacteria are essentially transparent, therefore it is necessary to stain them before they can be observed under the microscope. A bacterial smear is a procedure you do before you stain the cell. You add a drop of sterile water placed on a clean slide. You then add the bacteria to the water, and then the bacteria mixed in the drop of water and smeared all over the surface of the slide. Let me try and show. If this were the loop, the capsule that hopefully you can see the inside of the capsule, you wouldn't want to fill the entire loop full of cells. You'd only want to have enough cells in the loop that you could see the cells, meaning you have very little cells, something like about 1/10 of the loop full of cells, not very many cells, enough that you can see them. If you have a loop full, you have way too many cells. In fact, if you have 1/4 of the loop full, you'll have way too many cells. The slide is then allowed to air dry, and then the slide is heat fixed, which the organisms are fixed on the slide by gentle heating. This is so that the cells will not be washed off during the staining process. Any question about that? So the bacterial smear is something you do before you do the staining. Students always get that wrong. Simple staining is a technique using one dye to stain the bacterial cells. It will allow you to see the shape and the growth arrangement, but you will not see differences between different types of cells. Differential staining technique usually uses 2 or more dyes to provide additional information, such as you can get the difference between gram-positive and gram-negative cells. And then lastly, special stains are used to dye particular bacterial structures in the cell, such as the cell capsule, endospores, and flagella. When we look at cells, we can see different growth arrangements like streptococci, syngococci, staphylococci, tetrads, diplococci, and what am I missing here? Sarcsinia, 8 cells stuck together. And streptococci is right there. Seems like we're missing something, but I'm not sure what it is. All right. For bacilli, you can get syngobacilli, streptobacilli, and then diplobacilli. We do not have staphylobacilli. We do not have tetrads and Sarcsinia for bacilli also. The cells can be monomorphic in shape, which most species are, but they can be pleomorphic. A few species are pleomorphic. And then there's the spirilla, which differ from the cocci and the bacilli. The spirilla typically are only singlets. So that's the only cells, usually the cellular growth arrangement, usually we see with the spirilla. Now, don't be confused because I talked about shape, cocci, bacilli, and spirilla here. And then I also talked about the cellular growth arrangement. And that would be the singlets, the diplococci, the tetrads, the Sarcsinia, staphylococci, and streptococci. All right, a differential stain is a stain that usually uses two or more stains to give you additional information between different species of cells. And the cells will stain differently, why it's called the differential stain. There are two differential stains you need to know, and that's the Gram stain and the acid-fast stain. Shown down here someplace. There it is here. Acid-fast stain. The Gram stain was invented by Hans Christian Gram to distinguish between gram-positive bacteria and the patient's own cells, which were staining gram-negative. But microbiologists used this stain and realized it separated bacteria into two very large groups, the gram-positive bacteria and the gram-negative bacteria. And the difference is just whether the cell retains the crystal violet, the primary dye of the Gram stain. Bacteria that retain the crystal violet are classified as gram positive, and they will stain purple, as shown here. And then the gram negative cells do not retain the crystal violet, and they appear pink, as shown here and there. The difference in the Gram stain is based upon the cell wall of the bacteria. The gram-positive bacteria have a very thick cell wall with a very thick peptidoglycan layer, and the gram-negative cells have a much thinner peptidoglycan layer. They also have an outer membrane to the cell wall, which is a lipid bilayer. So the thinner peptidoglycan layer, and there you can see that's actually several layers thick, more than one layer. I don't know how many layers, but it's up to 20 layers. There's one, there's two, there's three, there's four, there's five, there's six, there's 7, 8, 9, 10, 11. This has about 12 to 15 layers of peptidoglycan. And there's the peptidoglycan layer in a gram-positive cell, and it's much thicker. No outer membrane in the gram-positive, outer membrane in the gram-negative cells. The decolorizing agent will destroy the outer membrane of the gram-negative cells. Shrink that down again. After bacteria have been stained with the crystal violet, iodine is used as a mordant, which makes a crystal violet iodine complex. Next, the cell is decolorized with an acid alcohol wash. It's applied only briefly to remove the crystal violet iodine complex. Gram-negative cells decolorize more quickly than gram-positive cells due to their thinner layer of peptidoglycan. So the timing of the decolorization is critical. Gram-negative bacteria are colorless, and to get them to stain, you need to apply the counter stain, which is usually safranin, which dyes the cells pink. Here is a mycoplasma, and you can see that this species of mycoplasma is staining both gram-negative, these cells here, and gram-positive, this cell there, that cell there, and that cell there. what do you notice between the gram-positive cell right there and that gram-negative cell there, that gram-negative cell there, that gram-negative cell there? What do you notice that differs between these cells? Come on, there's two things you should be able to see that differ. Well, if you're not going to state it, I'm going to just move on.
Can you point to the cells again?
The gram-positive cells are the purple ones. This one, that one, and that one, that one. The gram-negative are pink. And I asked you, what's that cell differ from that cell and that cell, and also that one being different. But that's probably the best. Maybe that's a good one there too. Both of those, actually.
Would shape be one of them?
No, the shapes are the same. They're all rods, bacilli. Any other guess?
Is it the arrangements?
No, I only talked about a single cell. You'll notice that that cell has a gap in it, like this cell is broken. This cell looks like a ghost of a cell, and that is both a ghost and a broken cell. Okay, these cells are probably dead cells, decaying, and that's why they're staining gram-negative, because this is the same species, but these are gram-positive and those are gram-negative. This is a mycobacteria, but it's probably old cells, and old cells do not stain correctly with the gram stain, particularly among the the mycobacteria, which should be staining gram positively. These cells, on the other hand, have a very dark, full cell wall, and you can see that there's no breaks in the cell. And that's because they're viable cells. And that's why they're staining gram positive. And the other cells are dead cells, why they look ghostly or broken. And that's why they're staining gram negative.
I have a question.
Yeah.
Not every cell that stains gram negative is dead, correct?
Probably not. It might be just a sick cell. But obviously, if the species is gram negative, then all of them will be gram negative. But I assume you're talking about the mycobacteria. I don't know because we are only looking at the cells, but that cell there looks intact and it might be viable. It's hard to say because you'd have to take it out and see if it grows, but it could be viable. Okay. But that one is definitely broken in half, and so is that one. And this one and that one look ghostly, meaning they look like a ghost of a cell. It's because the cell isn't entirely there. All right, some tips for doing the gram stain and preventing gram variable staining of bacteria is avoid using old cells. You need cells that are only 2 days old or less to do the gram stain. If they're over 2 days old, the gram stain may not work correctly. Prepare thin cells of smears. If the smear is very thick, it's difficult for the dye to get down to the lower cells. And then if they do get stained, it's more difficult to get the decolorizing agent to get down to the lowest cells and decolorize those lower cells. So you want a thin smear, preferably one that is a single cell layer deep, only one layer cells deep. Heat fix the bacteria onto the side after the cell has air dried. If you don't air dry the cells, then you heat fix the cells, you'll get the water to evaporate, boil, and that will create holes in the cell wall, which will may allow the crystal violet iodine complex to wash out of the cell wall when it shouldn't. and then avoid overheating the cells during heat fixation. Excess heat disrupts the cell walls, and you can always look at cells, and if they look like they're melted cells, you know they've been overheat fixed. Any question about any of that? All right, and then use fresh staining reagents. Do not over decolorize. The best thing to do is hold up your slide so you're looking at it at an angle, add the decolorizing agent, and then watch the blue rinse down. Once the blue stops rinsing down, stop adding your decolorizing agent. Any question about the gram stain? Oops, we'll talk more about the gram stain later. And then let's talk about the acid-fast stain. Some bacteria, mainly the species of Mycobacterium and Nocardia, have a waxy substance we call mycolic acid in their cell wall. Mycolic acid is actually one of the main molecules in the cell wall of Mycobacterium, meaning peptidoglycan. There's a lot there, but there's a lot of mycolic acid also. And so you can't say the majority of the cell wall is peptidoglycan, because the majority is peptidoglycan and mycolic acid. And I'm not sure in the Mycobacterium which molecule is more common, because actually both molecules are very large and they're very thick on the cell wall for the Mycobacteria. These bacteria, the Mycobacterium nocardia, are able to resist the acid alcohol decolorization step, and they also resist staining by the crystal violet. And we refer to them as acid-fast bacteria cell because when we stain them with an acid-fast stain, it gets into the mycolic acid, the cell wall, stains the cells, and then these cells resist the decolorizing step and the acid-fast stain. Initially, all cells take up the primary stain, which in the acid-fast stain is carbonyl fuchsin. It's a cherry red color, and then the cells are decolorized with acid alcohol. The non-acid fast stain loses the color, the carbonyl fuchsin, but the acid fast staining cells, meaning the Mycobacterium, the Nocardia, resist the decolorization and they retain the carbonyl fuchsin. You then stain the cells with the counter stain, which is usually methylene blue or brilliant green. And it will stain the cells either blue or green, depending on which stain you're using. At the end of the process, the acid-fast cells will stain red from the carbonyl fuchsin. The non-acid-fast stains will stain either blue or green, depending on the color of the counter stain. The acid-fast staining is a differential stain because it uses two or more dyes, two dyes, to selectively distinguish between the acid-fast and the non-acid-fast stains. So here's an acid-fast stain. Let me blow that up a little. The red cells are the acid-fast positive cells, Mycobacterium, and then the non-acid-fast stains, meaning the green stains, are not Mycobacteria. Any question about the acid-fast stain? So these are acid-fast staining negative, and then the red cells are the acid-fast staining positive cells. All right, let's talk a little bit about special stains. The negative stain is sometimes used to stain the glycocalyx, and that's because the negative stain stains the background of the cell, and if it stains the cell as well, it will show up the glycocalyx. Usually, a negative stain like India ink will stain the cell lightly, and the background darkly. Some negative stains use 2 or more dyes, and that would stain the cell and the background of the cell, but not the glycocalyx. If the glycocalyx is firmly attached to the cell wall and is a structure seen in the light microscope, we call it a capsule. And the capsule, if you remember, is less than 95% water, meaning water is probably the main molecule in the capsule, and it's actually the main molecule in the bacteria as well, because all bacteria and all life cells, living cells, are 70% or more water. And the capsule is probably 70% or more water, but it's less than 95%. The slime layer is 95% or more water, and it doesn't show up under the light microscope. Production of a capsule is important because it can be associated with an organism's virulence or its pathogenicity. The capsule helps protect the bacteria from being phagocytized by a white blood cell. If you're growing cells on a petri dish and the colonies look mucid or mucus-like this, that's because the cells have a capsule. Cells that have a capsule tend to be, the colonies tend to look mucoid like that. Capsules are not easily observed using conventional staining method. So like when you're staining with the Gram stain, it will wash away the cell capsule. Therefore, a negative stain technique is often employed to microscopically demonstrate the presence of a capsule. If you don't know a negative stain, usually you just add the stain on and then look at the cells. And so there is no washing step to wash away the capsule. Like here, we've just flooded the slide with India ink and then look at the slide with wet India ink on it to look for the presence of capsule. And India ink stains the cells light gray and stains the background a dark color, dark black. And then the capsule is staining or not staining. It's clear or white. On this slide here, which is a capsule stain or a negative stain, we are probably using 2 dyes here. One's that's staining the cells a dark color, and then a second stain, which looks slightly greenish gray, staining the background, and the capsule is unstaining. Any question about any of that? And that's showing you Klebsiella pneumoniae, which forms a capsule, and the body has a hard time throwing this bacteria off. Why, if it gets in the lungs, it tends to form pneumonia. And before the advent of antibiotics, pneumonia was a very serious disease because, especially if it had a capsule, the body would have a very hard time fighting it off, and the bacteria would just grow in the lungs until it tended to kill the patient. All right. Another stain is the endospore stain. It's a special stain standing for the presence of endospores. Endospores are metabolically inactive cells. They're hardy structures that are produced by some bacteria as a defensive strategy to survive unfavorable environmental conditions. So it is not a reproductive structure. It's a survival structure, and one parent cell makes one endospore. Why we don't call it a reproductive structure. The endospore is something that can survive harsh environments, and it just waits until the environment becomes favorable, and then the endospore will germinate, and the bacteria will return to its normal metabolically active vegetative state. And instead of looking like an endospore, it'll appear for the normal bacteria, most of the cells that form endospores are bacilli. There is one species of cocci, but most are bacilli. And so it'll change its shape from the endospore to a bacilli. And that'll be a metabolically active vegetative state. And this cell can grow, reproduce, it's metabolically active. The endospore has very low metabolism. It does not do much. It doesn't reproduce. It doesn't do much except for survive. And most endospores belong to a species of two genera, Bacillus or Clostridium. And those are the two main bacteria producing endospores of importance, which is shown here. The genus, Bacillus anthracis, and then the clinical significance causes the disease anthrax. Bacillus cereus can cause food poisoning. Clostridium botulinum can cause botulism, food poisoning causing botulism. Clostridium tetani causing tetanus, also called lockjaw. Clostridium perfringens causing gasgangrene and food poisoning. And Clostridium difficile, also called C. diff, causing antibiotic-associated colitis, also called pseudomembrane colitis. With C. diff, with the patient in the hospital, they can get C. diff, and to treat the patient, you have to treat them twice because C. diff can make an endospore which will survive antibiotic treatment. And what you do is you treat the patient, killing off the vegetative cells, but the endospores can survive the antibiotic treatment. And then you wait a few days and then treat with antibiotic again, and the endospores will now be in a good environment, so they'll germinate. You only need you need to treat soon, within a week. It's only a few days they normally treat, because the parent cell cannot make another endospore. If you wait too long, then you'll have to treat the patient for antibiotics like a third time. Any question about endospore forming bacteria? The Shaler-Fulton stain is an endospore stain that uses malachite green as the primary stain, and then heat is applied as a mordant to help the stain get inside the endospore. The cells are then decolorized with water, which removes malachite green from the vegetative and the perianthal cells, but it does not remove the malachite green from the endospores. Safranin is then applied as the counter stain, just to stain the clear staining cells after the cells have been decolorized. The vegetative cells then appear pink from safranin, and the endospores appear dark green from the malachite green. So here we have endospores. Let me blow this up. There's an endospore that's come out of the cell. There's one there. You'll note that they are Coccobacillus in shape. Sometimes they can be cocci in shape. This one is Coccobacillus. And here we have the endospore actually inside the parent cell. We have a little bit of the parent cell staining pink on that side, and a little bit of the parent cell staining pink on that side, and then the endospore staining green, and it's off on the side of the cell, but inside the parent cell. Any question about that? And that's why it's called an endospore, because this spore forms inside the parent cell. And when that parent cell dies, the endospore will be released. And then that endospore will just wait for the environment to become favorable for the species to grow. And then the endospore will germinate, and the endospore will change into a bacilli, and then will be metabolically active and can start dividing. Any question about any of that? All right, another special stain is the flagella stain. The flagella provide the cell with motility, and it's the only structure a prokaryote has if it is motile. Flagella is composed of flagellin. That's a class of protein. So flagellin is a protein that is found in the flagella. The number of flagella on a bacteria surface is useful in identifying and classifying bacteria. You don't need to know the names of this except for in this lab. If there's only, well, first of all, there can be no flagella, and then there can be one flagella on one end, monotrichous, multiple flagella on one end, lophotrichous, amphitrichous, multiple flagella on two ends, and then peritrichous, multiple flagella all over the cell. The flagella, when you look at the cells, they will look pleomorphic because if the cell is peritrichous, before it puts out all the flagella, it'll be amphitrichous. Before it's amphitrichous, it'll be lophotrichous. Before it is lophotrichous, it'll be monotrichous. And before it is monotrichous, it will have no flagella. Like after the cell just divided, it'll have no flagella. And given time, the cell will eventually become peritrichous, but that takes time. And when you stain the cell, some of them will be peritrichous and other cells will be some level of flagella underneath peritrichous. Flagella are too thin to be stained with standard staining technique, so special stains are required to view them under the light microscope. Usually they use a mordant, which applies thick layers on the flagella to make them show up. And here is the flagella stain. And as you can see, that cell is peritrichous. This cell has most of the flagella on one end, maybe not all of them, but most of them on one end. There's one that has cells only on one end. And if I look really, really carefully, I can find monotrichous, but I'm not seeing one at the moment. Oops, wrong one. That looks like it's got two. Maybe that one's only one. And that's all the same species, by the way. All right, know the terms of this lab? You don't need to look at the references. While you not be physically performing these activities, you must understand how to prepare a bacterial smear for staining and understand the bacterial smear is something you do before you do the staining. You should also understand the principles and procedures used in the various staining techniques and be able to interpret those results from a photograph using the microscope. So answer the following questions. And we're in procedure one, we're going to be preparing a bacterial smear for staining. There's a little video clip on watching how to make a bacterial smear. And there's another video clip showing you heat fixing. I don't know if it's showing you air dry, but you air dry the slide and then heat fix it. And there's the steps there. And then answer in procedure one, what are 4 common errors when preparing a bacterial smear for making a stain? So you need to state 4 common errors. And then procedure 2, we're going through the simple stain. View a clip, a video of a simple stain. It's adding 1 basic or negative dye. It doesn't need to be a basic dye, just 1 dye. And then you wash off the dye, let the slide dry, and then look at it under the microscope. And all the cells should stain the same color, but you can distinguish different shapes. The first question will be, what is that, procedure 2A? Let me come back here and procedure 2, question number 3A. First question is describe the cellular morphology, 1 of E. coli. I'm going to tell you it looks like it is a spirochete right there, but that's because there's 2 cells stuck together. E. coli is not spirochete, and that looks like a spiral there. Those are more 2 cells, 1 cell there and 1 cell there. And then tell me the cellular morphology, that's the shape of Staphylococcus aureus right here. And then procedure 2, question 3B, give me what's the total magnification of both of these slides here. Remember, the total magnification is the objective lens magnification times the ocular lens magnification. In procedure 3, we're going to go with the gram stain. We have a video clip to doing a gram stain, and we have a virtual lab on doing the gram stain. Let me briefly open the virtual clip just to get to the lab. It may take a little while for that to download. I'm going to just quickly go forward because I want to show you something.
This is what. Now take a look. This yogurt. In the gram state bacteria. We're going. All right.
You can go through that.
To prevent bacteria, you're going to use a striker to light the Bunsen burner. We need to dilute the. Start by placing 1 milliliter of the diluent. This is an inoculant. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. After it has cooled down, place the yogurt sample into the Now, sterilize the inoculating loop again in the flame of the Bunsen burner. Transfer a small amount. Place 2 loops full from the sample onto the slide.
Trying to get to the point where I want to show you. I mean, obviously you need to learn this
Tilt
stuff
the
too.
slide to spread the dropout slightly. Now we'll heat fix this. Place the slide in a class, such as a clothespin, and pass it over the flame of the
That
Bunsen
should be
burner.
after the slide air dried, by the way.
Now we begin the staining. We use 4 chemicals to stain a slide.
All right, so this is
Apply
the
them
start of the
in the
gram
corner
stain.
And rinse the slide off under slow running water in between each chemical.
So this is telling you we're going to wash after each of the 4 steps of the gram stain.
The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria.
All right.
Using the dropper, squeeze out enough drops to cover your sample.
So this has crystal violet in it, and so what you do is the arrow tells you what to do.
For 60 seconds.
Go 60 seconds.
Now rinse it under slow running water. Now use the bottle of Graf's iodine.
I just wanted to show you how you work this, okay?
Let that sit for 60 seconds.
All right, any questions about how that works? Oops, I didn't mean to stop the share. Let me share that again. I want to stop this video. There we go. So do the virtual lab. It's the only procedure of the gram stain you will actually do. And then review the steps from performing the gram stain. First, you flood the slide with crystal violet for 1 minute. Then you rinse. After each step, there will be a rinse. I won't talk about the rinses any further. Second, flood the smear with iodine. Let it soak for 1 minute. Third, flood the smear with acetone alcohol and let it soak for 5 seconds. The best way is to view it and then watch the crystal violet run down. Once it stopped running down, stop adding the crystal violet. Now in our procedure here, we are using alcohol acetone decolorizer. In the virtual lab, they are only using alcohol. So they are applying the alcohol for longer than 5 seconds. You need to apply alcohol alone longer than 5 seconds. You do about 20 to 30 seconds. And it's because alcohol does not remove the crystal violet from the gram-negative cells as quickly as alcohol acetone alone. I'm just pointing out that this procedure is a little different than the procedure in the virtual lab. That's all I wanted to say. And then fourth, flood the smear with safranin, it's the counter stain, and then blot the slide dry with bibulous paper, and then view it under the microscope. In the steps of the gram stain, after you apply the crystal violet, the cells will be blue, purple in color. After you apply the grams iodine, they will be purple in purple color. And you can kind of see this color looks different than that color, but these look the same. That's just the way the artist has done it. These do look a little more purple. Then you apply the decolorizing step. The gram positive cells will be purple color. The gram negative cells will be clear. Then you apply the safranin. The gram positive cells will be purple color. The gram negative cells will be pinkish color. Any question about that? Now remember, the safranin is getting on into all the cells. We just do not see the safranin with our human eye. And that's just because the purple is a more intense color, and that's the only color the human eye sees. With the electron microscope, you could see different colors. Complete the following chart to answer this question. The gram reaction, meaning what's the stain of the gram reaction? Is E. coli gram positive or gram negative? And then is it Coccus or Bacillus? There is E. coli, Staphylococcus aureus. The gram reaction, is it gram positive or gram negative? And is it Coccus or Bacillus? This is the Staphylococcus aureus. And then for procedure 3, we have procedure 3A, B, C, D, E, and F. These are questions. Staphylococcus aureus, after the primary stain is added, is it pink, purple, or colorless in color? E. coli, after the primary stain is added, state its color. Staphylococcus aureus, what color is it after the alcohol acetone step? Pink, purple, or colorless? E. coli, after the alcohol acetone step, what color is it? Staphylococcus aureus, after the counter stain is added, what color is it? And then E. coli, after the counter stain is added, what color is it? Any question about any of that? All right, let's go through procedure 4. It is going to be the acid-fast bacteria stain. It's abbreviated AFB stain for acid-fast bacteria. View the following video clip on the acid-fast stain. And there are 2 acid-fast stains. One of them that we're going to talk about is the modified Kenyon acid-fast stain. Both of them use both acid-fast stains use carbol fusion as the primary stain. It stains all cells pinkish color. You then apply heat, which is sort of a mordant. It allows the carbol fusion to more easily stain the acid-fast bacteria. You then do the decolorizing step. With this stain, the decolorizer is simply water. Water will wash the carbol fusion out of the acid-fast negative bacteria, but not out of the acid-fast positive bacteria. And then you apply brilliant green as the counter stain, and it will stain the acid-fast negative cells green, and the carbol fusion will stain the acid-fast positive cells pinkish. Okay, any question about that? Here are 2 smears below showing you acid-fast bacteria and acid-fast positive bacteria and acid-fast negative bacteria. And you need to interpret whether the organisms denoted by the arrows or acid-fast bacteria positive or acid-fast bacteria negative. So each arrow should have a discussion. And if you can't put the name or the type of bacteria by the arrow, then just stick it someplace after or before the picture. Okay? And you can discuss it. The leftmost cell is, tell me what it is in the first picture, and in the first picture, the rightmost cell is. On the second picture, tell me the leftmost cell is, and then tell me what the rightmost cell is. Any question about procedure 4? Make sure you answer the questions or it'll be wrong. Not the question, answer the arrows in this case. All right. Oops, went a little too far. Procedure 5 is looking at special stains. View the video clips for making endospore stain and a negative stain looking at the cell capsule. So for figure 5A, we have an endospore stain. I just know that. And for figure 5B, we have the flagella stain. And then for figure 5C, we have the capsule stain. Figure 5A goes to question 2A. Excuse me. Figure B goes to question B. And figure C goes to question C. Any question about that? In figure A, which is 5A, it says this is the Schaefer-Fulton stain, which uses what dye as the primary stain. So tell me the primary stain. And ultimately dyes what cell structure, which is produced by some bacteria to aid in survival. And then for, that's 5A1, for 5A2, list some examples of pathogenic endospore forming bacteria. And you can get that from the lab module. By the way, I should not be going through the questions in the lab module. I should be going through the questions in the worksheet. So let's switch to the worksheet. Looks like I didn't open up the worksheet for that one. Hopefully I stated that question. And then what are some examples of pathogenic endospore producing bacteria? You can get that from the lab module. For figure 5A, this slide is a wet stain that coats long whip-like appendages which aid the cell in motility. So what stain is that? And state the flagella arrangement type. So take a look at the flagella arrangement of this cell, and I should say of these cells, and state the flagella arrangement. And then what are some examples of clinically significant bacteria that produce flagella? You can once again get that from the lab module. Let me remind you that the flagella arrangement will be pleomorphic, and you should only name the highest flagella arrangement that you see. Figure 5C goes to figure C. This slide is a wet stain that uses India ink or nigrosin and can be used to detect the presence of wet cell structure, which is located exterior to the cell wall and prevents phagocytosis. So that would be C1, and that would be C2. And then C3, what are some pathogenic microorganisms that produce a capsule? And you can get that from the lab module as well. Any questions on those questions? In the case study here, case study 1 is a 45-year-old female experiencing respiratory issues. Based on her clinical presentation, the doctor orders a chest ray and a sputum for AFB culture. That's acid fast bacteria culture. In addition to the culture, a AFB smear is made and stained, shown here. And based on the findings below, what is the most probable diagnosis? You must name the species causing the infection. If you name the disease, you'll only get partial credit. Right there, we can see lungs. And if you don't know, the lungs should be totally dark, like this area here. And this is not a typical lung structure. That's not typical lung. This whole region of the lung is not typical. And then this is the sputum smear and stain with the Zeal-Nelson stain shown here. So give me the species causing the infection. In case study 2, we have a 64-year-old diabetic patient admitted to the hospital with soft tissue necrosis shown here. And you can see that's really ugly soft tissue necrosis. A routine culture and gram stain is ordered. The bacteria grow aerobically. Based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. There's the gram stain. The gram stain is shown here. And there's the Schaefer-Fulton stain. And you can see a structure in this stain. You must recognize that structure to get this species. You must name the species. If you simply cause the disease, in this case, it'll be totally wrong because the disease would not name the species. Any questions about any of that? All right. So work on the lab. I will be here until 9 o'clock to help any student with questions, but I will log off once the last student logs off. So make sure you ask any questions early. Obviously, if you're on the lab, I won't log off, but if you log off and then come back to ask a question, I may not be here if every student is logged off. Any questions about that? Well, that didn't work. All right, let's not end it, but stop the share. Stop the record. All right, let me remind you, you only should be submitting the worksheet for the lab. If you submit the lab module, you get a penalty because if everybody were to submit the lab module, my Canvas website would become full and then you students would no longer be able to submit your assignments. So please only submit the worksheets.